The only thing we do with the coefficient is to calculate this and this, the beta 0 and beta 1. Well, we're not calculating, sorry, we're accessing it. So, and as we said, this value is just the average of the expression in controls. This value is our fold change from beta 0 to beta 1. So where we ended up in here, right? That's what we did. Visualizing the coefficients. Again, this is not a step that you're going to do in your analysis normally. You're just, we're just exploring the data, right? So you get familiar how, if you need to access, you could. Hey, I'm using the mic, right? Okay. Should I get it closer? What about now? Better? No? Good? Should I speak louder? Okay, I speak louder. I blame you if I'm losing my voice already, and it's only Wednesday. Yep. So again, if I'm going too slow and you want to read, you can keep going. You don't have to wait. But I'll go step by step. So this is another thing, another complex thing that you don't need to know, especially if you're dealing with the most basic analysis, which is just comparing within a condition to treatments. But internally, the linear models, you use this matrix that you see here, that it's going to be hard to show in a single on the screen. I'm going to make the text. Sorry for the people in the back. Yeah. So internally, this is another way to look at the formula, right? So this is how your grouping is working. So actually, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 samples, which are our normal samples. And basically, internally, R is reading this matrix and saying, OK, how I calculate the intercept? What samples are the ones that I'm using for the intercepts? The ones that have a 1 here, these are the ones that are normal. And you are not using them. We have a 0. You multiply by 0 to calculate the coefficient. Now, OK, how do I estimate where beta 1 ends? So we scroll down. OK, these samples, they use 1 arrow plus a second arrow. These are the ones that belong to our condition. Now, again, this probably you're not going to use it that much. I'm showing it. Again, this is a simple thing. You only have one condition, normal plus treated. You don't need to reach these details in your analysis. But if we have a really complex model, and if we have time, I'll show you. This helps me, at least helps me, how to draw those arrows and understand how many coefficients D-SIG2 is going to calculate. So if you want to know how internally it's calculated or access the model matrix, it's through this function call. OK, D-SIG2 recommends some pre-filtering of the data. So we said 60,000, right? Ensemble, something like that, between 60,000 and 70,000 genes annotated. So if you do a volcano plot like Bonnie did, you're going to have 60,000 dots. And it gets really crowded. So one thing you could do, you could remove from your data genes that are very low counts, which D-SIG2 is not going to use anyway because they're really low counts. So for your visualizations, you want to remove dots from your data because there's going to be a lot of genes with 0 counts and things like that. You could remove them. That's one thing you could do. So this is one of the ways you could do it. So basically, it counts how many samples per row, right? So your samples are columns. How many samples they have more than 10 reads? If you have, in this case, I'll limit it to 10. If you have at least 10 samples with more than 10 reads, you keep that gene. If not, you throw it away. I don't care, because there's not enough information for me to do the analysis anyway, so I'm not going to use it, and you throw it away. So this could be a way to do it. I'll show you because this is one of the recommendations they do. I don't do it. Why? If we do this at the very end in your analysis, when you get your table, those genes are not going to be represented, which is fine. But sometimes I get questions, oh, what is SHANK3? I'm making this up, right? SHANK3 is the gene we're dealing with. I don't see it in my table. Well, my answer is I filtered it out, right? If I don't filter it now, it's going to show up with zeros. But at least the investigator knows that the gene doesn't have any information, so I know why it's not present. But it's up to you. We can do it or not. It's fine if we do it. Anyway, we are not searching for a specific gene, so we can do it. So we went from, in this case, we actually have 20,886 genes. We went down to 15,248. Actually, internally, and I think I give some details about it if I remember correctly at the very end, internally, DSeq2 has a way to remove unimportant genes because they have low counts. So you don't have to make a decision, arbitrary decision of saying, I'm removing these genes because they have low counts based on an arbitrary number. DSeq2 uses different approaches, and you can even select what approach you want to use to remove genes that are not represented enough. So internally, DSeq2 deals with it. Okay, quality control. This is another recommendation, which is to reduce the effect of the variability when you have low counts or high counts, right? So as you can imagine, if you have low counts, the variability is going to be higher across all your samples, replicates, and all that. So there is one transformation that we can use. There are actually those transformations, right? So this is called the variant stabilization or the R-log transformations. This is quite common. You also use these types of transformations in other fields, in single cell sequencing and all those. So these transformations are only used for visualizations, basically. When you do your heat maps or things like that, it's good if you transform the data first. And what it means is now we're going to have our original data and our transformed data. So we're going to have two counts. So we have the normalized counts that is being used for analysis, which the analysis is already done. And we're going to have our counts being transformed with one of these two systems, the variance stabilization or the R-log. Which one I'm using in this case? I think I'm using the R-log transformation, yes. And you can use the BST. So if you want, you can comment out these two lines and use the other one and see what happens. You can see if you notice a difference. Again, this is only for visualizations. I think the DSeq2 recommends VST when you're dealing with a lot of samples. We are not. So any of the two should work. And then we're still doing the exploration data analysis, right? We are not dealing with p-values or anything like that. Everything is already being calculated. We're just exporting the data. So like in PCA, so the next step is going to be our principal component analysis. Instead of using the original data, the normalized counts, we're going to use the transformed counts. And again, the main goal is to remove this variability so the variability on lower counts is going to be much higher, and the variability in higher counts can be also higher. So we're trying to reduce the effect of those and focus on the main genes that are in the middle, which are probably the better, best. It's working for you guys? Still thinking? Well, another way to know if it's busy or not is if you see a full dot, I think it's thinking. And also you have it here, like it's working. Let's give it a minute. Yep, it seems, I mean, we're keeping the CPU busy, so I'm assuming it's doing the calculations. So meanwhile, should we review the principal component analysis, what it means? That's the idea of dimensional reduction. So we have 15,000 dimensions, so this is the number of genes, and we are trying to represent the same information in a plot that only has two axes, right? PC1 and PC2, for example. or 3-axis, because I'll show you how you can do a 3-dimensional thing. Sorry, today AWS only allowed me to choose a relatively small computer for whatever reason. So that might be the reason that it may take a bit longer. Questions? I feel uncomfortable being in silence. Can you? Well, first, this needs to finish. Did it finish? It may take time. Again, if you see the asterisk here, it means it's working. If there's nothing wrong with the website. So until you don't get a number, we should get a number that is after whatever number we had before. Then the PCA. I mean, you can execute. Actually, you could execute the whole thing, and it should work step by step. Except that sometimes you need your input. So, but once you fill all the gaps in the whole notebook, you could execute the whole notebook at once, and it's going to go step by step. For whatever, I don't remember being taking that long, but it could be. If for somebody finishes, please tell us so we know that it's working fine. And oh, it finished for you? Oh, okay, then we only need to wait. Okay, that's easy. So for plotting PCAs, I'll show you 2 ways. This is the internal function from DSIG2 to create the PCA. And we're going to use a manual one, or the R1 way, basically because I thought it's easier for me to access. You remember the script plots to know how many, so the percentage of variance of explained by each PC, for me was easy to access through using the standard way of R. But you can, if you only care about a plot, simple plot, you can use the one provided by DSIG2. Did we get results for anybody else? Only for the smart ones over there? Are we saturating AWS? I think AWS is smoking now. Yeah, it's 100% everything, okay? Yeah, we're reaching the limit of the... So if you keep reading, please, exactly, read the next section, so when we reach it, it's going to be easier to understand. The one about principal component analysis. And have your questions ready, if you have any. So somebody yesterday was asking me, oh, what if we reach the limit of the CPUs in the system? A lot of people using it. I think we have an answer now. So if you're curious, we're currently using 32 CPUs. So we have 40 people requesting a job. So definitely the computer is on its limit. You have questions? Did it finish for anyone else? How did you do it? You started earlier, I guess.
No, we did at the same time. It was
Listen.
just luck, I guess.
Okay. Is any of you a stand-up comedian and wants to come here and explain some jokes while we wait? Or anyone who wants to hear a story from Bonnie, the long ones? You're too quiet today.
I'm finishing some of my work.
Oh, that's what it is. That's why you're quiet. And you are not interrupting and saying something that I already said in the previous slide?
He's banking.
Is,
He's banking. No, actually, Francis.
yep.
So we have to actually ask them a question. Like, so you all are doing this transformation like VST and R-log transformation. Why you need to do transformation? Why you need to transform data?
I guess it's more genetic than what I said before because you
Yeah,
need
no,
to.
I mean, I don't know whether they really appreciated that what you were talking about dispersion. That is why I thought that we'll
I
add
think
some.
that's an insult, but yeah, go ahead.
No, like, so what do you think? Why you do?
The compressing by log transformation.
Yeah, I mean, why you need to do that? Why?
Sorry, this is not what I want to open.
So many large numbers will be flashing.
No, I think he said that we are converting the outliers.
Yeah, but I think he wants a more generic answer than the specific. He doesn't care about if it's R-log, VST. You're talking about general transformation
Yeah,
of data?
general. So it doesn't mean that. So people do all kind of transformation. They have statistics and things like that. But what we are really, why we need to do that? I mean, we have a data, we captured it. We just put everything there without any change. But why we need to transform that?
I mean, the large big numbers count. So we are giving a smaller number by log transforming, compressing, and we bring in more uniformity.
More clear image visualization.
Or better comparison.
So actually what happens that in most of the data sets, that when you have the gene expression level, how much actually gene expression, lowly expressed gene have higher dispersion. And then, so what happens really, you want to actually control the dispersion in your data. So you are, by transforming, you are actually pushing that low expressed gene further to the line. And this is log for that. So essentially what you're doing in all this, whatever
Yeah,
you are
the
doing,
battery is
VST,
gone though.
log, or any other, let's say, other kind of normalizations, if you have, there's a linear model
I kill it.
so all this kind
He
of
deserves it.
normalization, the idea is that can you control the dispersion in your data in a such a way that the gene expression itself should not actually impact, the level of gene
It's
expression
hard
should
again,
not
right?
impact your differential gene expression or significance. That is actually the idea here. Controlling the dispersion in a way that the lowly expressed gene or highly expressed gene should not influence your significance calculation.
In this case, the transformation is only for visualization. The transformed data is not used for analysis. The analysis is already done. We already done all the analysis. Sorry, yeah, I dropped it and I lost the battery. It's around, it's preloaded, yeah.
No, refill that in the bag.
How long it did took only 1 minute or two for you, right? So one thing I'm gonna do you mind to, I don't know, should we try to half of the class stop it and the other half run it and see what happens?
Yeah, no, Alex, what we can do, this is like 11:15. Francis,
Yep.
let them run, and I think I will actually let this keep running. I will actually start the NGS class, and so that will give them some time to finish all of it.
Well, we hope so. And it's not stuck for whatever reason.
Let's see. Okay, so let's do that way. I have sent you a presentation. Can you?
Oh, it works even here. Yeah, that's cool. You can scroll up and down.
Uh,
Well, let's get ready for Bonnie then.
come, come. You have to start. Okay. Yeah, just put those slides somewhere, anywhere, and then I will rearrange it. Anywhere, anywhere, just put it. I will, or will. Okay. Yeah, just before clinical application. Yeah. Yeah. Okay. Yeah.
So while your code is running, just a few things. So when we think of transcriptomics, there are three kind of layers to it. Bulk transcriptomics, single cell, and spatial. And the widely publicized analogy of a bulk is like a smoothie. So what you get is that you don't, I mean, in a smoothie you put everything. You put strawberries or whatever fruit of your choice, bananas, and you don't know what taste is coming from what. So the analogy of a bulk transcriptomics is to a smoothie, whereas in a single cell, it's like a fruit salad maybe, where you know exactly, if you want to taste, you know how does a kiwi taste, how does a lemon taste, or a berries or mangoes or whatever. And in spatial, you also have information not only about each single cell, but also about the spatial orientation of that cell. And it's widely used in pathology actually to know about the single cell spatial transcriptomics. So in terms of immunology, when you look at the different uh, ways that you can employ single cell genomics or transcriptomics, you can look at the different antigen receptors, like you can do a T cell receptor or B cell repertoire analysis. You can look at cell dynamics, you can look at the interaction between cells. You can look at cell-cell communication. And if you're doing single cell spatial transcriptomics, you can also look at the tissue architecture and the cell-cell communication. And in that way, you can dissect the heterogeneity within a sample which would appear to be homogeneous. And the essence of single cell transcriptomics is similar. So you first do a sequencing, you get the reads, you align the reads, you do a quantification as you do in bulk, and then you do QC steps. One of the QC steps is to look at the reads coming from the mitochondrial genes, because you don't want that. I mean, there should. So one of the QC steps is that you should not have too much of mitochondrial gene expression in your sample, because it indicates that a poor viability of your sample. So what is the threshold that you generally use for this, like for mitochondrial, or do you filter out on that?
Yeah, we usually filter out. I think Francis can be more doing.
Well, it depends on the sample.
Okay.
It could be 10%, could be 20%.
Or you can even relax that? I mean, even 20% would be good enough?
I mean, depending on your situation,
Okay.
you need to deal with, you're looking for for a population that is very low in your sample, and you need to keep as many as you can. You probably need to relax the frequency of filtering.
Then you do a normalization, as Francis talked about, and then you can do clustering. So you can see that different cells, based on their gene expression, do they form distinct clusters. But apart from gene clusters, clustering according to gene expression, you can also look at trajectories, or linear trajectories or based on the gene expression, which cells are rising from which progenitor cells and all that. So you can do a trajectory analysis based on your single cell data. You can also look at regulatory networks when you have single cell data. So these are some of the applications from single cell transcriptomics. You can look at clusters, like are there any distinct clusters based on gene expression. You can look at trajectories, what cells are derived from which cells, are they related to each other in some way. You can look at regulatory networks, and you can also look at cell-cell communications. And in terms of spatial transcriptomics, so this is from from a kidney sample. So in a single transcriptomic map, you can have a spatial reconstruction using either the landmark genes, or you can do it de novo. And then you can also look at cell communication networks. So in this case, you have got like pelvic epithelial cells from the pelvis of the kidney, neutrophils and macrophages, and you can look at the different cytokines or chemokines which are used to communicate between these cells. And the lower figure, you can see the nephron, which is divided into three parts: the cortex, the medulla, and the pelvis. And different fluorophores are used here: phalloidin, Lis-M, and a red fluorophore for CD11B. And so, based on this, you can look at cell networks. You can do a spatial reconstruction. This was actually a transgenic mice which had a susceptibility to pathogenic E. coli. And so the Lis-M was that mice, and the merge shows that where the CD11B and the Lis was, that shows off as yellow, and the phalloidin in the cortex and the medulla is showing as white. So in spatial transcriptomics, there are different methods to do it, different platforms to do it. So, but that you can do it. The single cell spatial transcriptomics can also be captured at a single cell resolution. So I think that's what I wanted to talk about. So you will be.
Yeah, I will be talking more about the spatial transcriptomics as we finish this talk class. So this will be the last one for the NGS technologies. So we were actually yesterday we were discussing about the applications of NGS, and one of the very recent application is proteogenomics. That transition, slide transition. Oh, okay, thank you. Okay, so the slides got a little shabby when we are actually transferring it. Okay, anyway, so one of the way to do this, there are two different approaches how you will do the proteogenomics. Okay, so proteogenomics stands for studying protein or quantifying protein, or both identification and quantification of protein using genomics. So till this time, most of the protein identifications are done through mass specs or low res ELISA kind of a technique.
but there were no way to do like thousands of protein assays on hundreds of samples together, and the proteogenomics is actually opening that window. And there are two different ways to do this. One is called as SomaLogic, or which is a company called as SomaLogic, which is recently purchased by Illumina. And sorry for being this bad slide. Actually, it was not like this when I made. So in case of SomaLogic, what they are doing, they are using something called as aptamers. So aptamers are DNA molecule. Okay, they are actually nucleic acid, and those nucleic acid can recognize proteins. So essentially, what is happening that the DNA or nucleic acid makes a tertiary structure, and that gives you a binding pockets. And the base, you can change the sequence, and you can actually change that binding pocket where protein can specifically interact. So that is a very specific. So interaction of aptamers with the protein is very specific. That means you design aptamers unique for each protein, and that will give you, it's kind of an antibody protein interaction. But the good thing about aptamer is that antibodies are still sloppy, but aptamers are very, very specific. But then the problem with aptamers is that it needs a lot of designing and validation. But once you have an aptamer, then its specificity is amazing. So it's not as sloppy as. So this is an aptamer. They have some modified bases and normal base. And in this reaction, what you do, this aptamers, it has a biotin molecule here, a linker, and I will tell you why all of these things are, and a fluorophore. So NGS based method, you don't need that fluorophore. They are there for microarray based. So this platform first came for microarray. Now this is also available for NGS where this fluorophore is not used. Now what you do, you immobilize these aptamers on a streptavidin bead. So these are biotinylated aptamers. So you immobilize them on a streptavidin beads. So once you, and then you add your protein, mixture of protein. So if it is urine or plasma, whatever it is, you don't have to isolate protein. You can actually add the real material like plasma or urine or cell lysate. I mean, you can't do cell lysate with SomaLogic, but with the Olink you can do that. So you add them, so protein will go and bind to its specific aptamers. Those are immobilized on a streptavidin beads. Now you pull everything out, you wash anything that is not bound, any protein that is not bound, you wash them off, and now you have a very specific interacting aptamer interacting with protein with all this, linkers and all those kind of stuff.
Why didn't it work for a protein lysate?
Hmm?
If you took the cell lysate,
Yeah,
they extract the protein. It will work for that.
yeah, it will, but the whole idea is not that. The problem here is why we don't do that, because the assay, these are like high content assay. You want to analyze thousands of samples in a go, hundreds of samples in a go, or something like that. And second thing is that you don't want to use a lot of material. These assays are done with 55 microliters of plasma. So think about like extracting, the patient samples are scarce. You don't get lot of material from the patient sample. So they want to actually use it as minimum as possible for this kind of assay, and then only this will work. So that is why they don't go to that path of extraction. It will work with extraction also. Okay, it's not that it won't work, but the idea is that you don't have to. If you do extraction, you have to spend extra money for extracting those things also, no?
But if you are comparing a cell, if you have a cell where you have the transcriptomic data and you want to compare the protein data as well for that cell, then wouldn't it be a good idea to have the protein from that cell?
Yeah, protein from that cell also. Like you take the cell, lyse it, and so lysate is that protein. You don't have to purify in that case. So you can work with lysate. So that is.
We are doing one project where we have transcriptomic data for some patients with SLE. So where we have done the transcriptomics of the PBMC, and then we thought that protein will go on the serum or what, but then it's not the same.
Yeah, so just lyse the PM.
That's process we are doing.
Yeah, we can do that. We can do that. So these are now those interacting DNA and somamers, what they call as, they are actually now immobilized on a streptavidin beads. So once you do that, after that, what you do, I told you like there is a, if you remember this, there is a linker here. This linker, what you do, these are photocleavable linkers. So you shine UV light on that, and then it will cleave your complex that is actually bound on streptavidin beads away from streptavidin beads. So now everything is in solution. So, you basically remove the beads and you take your solution out. And solution what you have is actually this complex, which is protein aptamer complex. Okay. Now, you add A, and so now the other thing is, during this step what also you are doing, you are dissociating. When this cleavage happen, you are also dissociating any protein that non-specifically interacted, you know. So, there are some low intensity, low affinity interactions are also there, which is non-specific. So, that they will be actually during this cleavage, they will assure that those interactions lose the contact with each other. And so, now the non-interacting proteins and your aptamers now separated, but those one which are specific will stay in the complex, and they will add something called as a polyanionic competitor, which will go and bind to the free protein. So, that means now these free proteins are not, they cannot come and bind back. So, non-specific proteins which are in the solution, which are free of aptamers, when you add these polyanionic competitors, they will come and make a complex, and they will stay away from any further interaction. And that means now you have a very unique complex which will with somamers and
How these non-specific are released?
eh?
How these non-specific proteins are released?
So, by when you are doing this, when you are actually really cleaving it out of your streptavidin beads through the linker dissociation, in that step the salt solutions are also changing a little bit. So, then that will actually release any non-specific interaction, and non-specific protein will come into the solution, and their respective somamer also will come in solution. Then they will add a polyanionic competitor that will go and bind with the free protein whatever is the solution. And then what you do, you actually go and add next biotin moiety into the complex, the aptamer protein complex, and you pull it down. After that, you dissociate the protein and aptamer, which leaves a set of a DNA fragment. So, basically now you have a set of DNA fragment, you make library and just sequence it. So, those and each of those fragments, short fragments of aptamers are corresponding to one individual protein, and you just do like RNA-seq kind of analysis, where you say, okay, you count how many of those are coming from each of those molecules. So, you are generating essentially same thing what Francis is teaching, that now this is not RNA, but what you are doing, this aptamers, you count how many, what is the count of those aptamers in your sample. And no, it is real quantification of protein. Yeah, relative in sense like it's all, they have always a standard curve, okay, and on that actually the quantification comes in. So, in that way relative means like you are actually benchmarking it against something, but that is not really they are doing. So, this is how it is actually done. So, you have somamers interact with your protein of interest, then you somehow think about all these things you are doing, and then finally you get that complex uniquely, and then you get the DNA out of that, and then make a library and sequence it, and then you count it, and then. So, there is another technique called as O-link, which actually Dr. Amit was mentioning.
You want to examine multiple proteins?
Yeah, so SomaLogic can analyze up to 11,000 protein in a single lysate. So, as 55 microliters of a lysate, you can analyze up to 11,000 protein. You can.
We need different type of aptamers.
So, yeah, it comes as a mixture. So, it will come like 11,000 aptamer mixture. Yeah, they have customs also, but yeah, but the highest size is 11,000. So, it comes as a mixture. So, you take the mixture, you add into your thing, and then everything happens. Yeah, 11,000 is the maximum. In case of this another company called as O-link, which also makes a proteogenomics kit, and this is a little more simpler than and it's antibody based again. So, any problem that you can think of antibody is there in this case, but the way how they get specificity. So, what they do, instead of using one antibody, they use two antibodies. So, and then each antibody will have a small DNA fragment attached to it, which is complementary to each other. Okay. If actually random antibodies comes and interact with it, they won't make the complementary pair. So, if the right antibody comes and then recognizes two different domains of that same protein, then this will make a portion of that DNA will be double stranded, and they just extend that like primer extension you do in case of, and you extend that and make it whole completely double stranded molecule, and that double stranded molecule now you have this double stranded molecule, you just make a library and sequence it. So, each of those again sequence can be traced back to the antibody, and then antibody can be traced back to the protein of your interest. So, this is actually, this is how two different approaches, one is based on aptamers and one is based on antibody. Yeah.
Even in the Olink, they have basic versions where they're using microarray.
No, Olink don't have any microarray based.
For the smaller numbers, they have a microarray, hybridization based.
So not microarray. So they have they are called as Q100 system, which is based on qPCR. So what they are real TACMAN based qPCR probes. So once you release this DNA, now how you quantify it's up to you. Okay, so one of the way they do for the smaller panel for 48 panel, they are using something called as old fluidine super arrays. So those who are actually old enough, they know something called as fluidine super array. Super array means what happens that you have, it's a qPCR. So you have a 96 well this side of it, it's a glass plate, and 96 well this side and 96 well this side. And in this 96, this side of 96 well, you will add samples on all the 96 wells, and this side you will add primers with TACMAN primers. So you can add all individual 96 primers. Then you spin your glass slide. What really happens that there is a center, there is a microfluidic chamber is there. So liquid will flow in all the combinations. So you will get combination of sample. So 96 by 96 combination you will get, and then so that means you are doing that many assays. And you will, and it will do the qPCR, and then you calculate the CT values and do the quantification, and so the what Olink is using the Q100 system, they are using something like that. So it's a modification of their old fluidime system. But
So if this antibody which have DNA molecule labeled to them, so can this single stranded DNA annihilate the complementary strand without the antibody binding with the antigen?
no, because of actually so many molecules are there that it's hard for them to, because so DNA affinity is it's based on affinity. Okay, so DNA affinity in a liquid system is actually lesser than antibody protein affinity. So any reaction kinetics is actually driven by that affinity of a reaction. Like so whichever is having a higher affinity, they will actually reaction will tend to actually go to that direction. Okay, so it's not that free DNA cannot actually do a reverse complementary. It's just because the favoring reaction here is antibody protein reaction, and so that is why most of the molecules will be
Still there may be some free antibodies available for
less. It's very less. Actually, it's so crowded environment that actually it's very less. That is, and then the salt optimization and all will give you that balance, and that is the innovation here actually.
They call it
Proximity ligation, yeah, that is what actually it is called as.
maybe complementary bases are less.
No, it will not, very few. That is the innovation there. Okay, that is how the whole reaction is actually optimized in a way that just antibody based pairing should not happen, and that is done through controlling the salt concentration in the solution. Okay, so we'll see a little bit more time anyway. We are, if we could finish it, then okay. Anyway, so one of the major application, and why all of us are here, because we are interested to use NGS for the clinical application. So there are various type of variants you have which actually can be identified through NGS based approach. Like single morph SNPs are there, insertion deletions, repeat expansions, frame shift mutation, defective splicing, or even the premature codon insertion in the genome, okay, in the genes. So what is actually the purpose of DNA or precision medicine, DNA sequencing and precision medicine? It can be for discovery, it can be for diagnosis, it can be for classification of disease stage, it can be for prognosis, it can be for therapeutic choice. So it depends upon what is your application. You can actually tailor your NGS based analysis for, but the problem here is that sequencing a genome is pretty simple, and and as I mentioned you, like we are sequencing every day and 30x genome, almost 200 250 genomes every 48 hours from our facility. So sequencing is not a problem. It's simple, and now it's not expensive too. So that means simple, inexpensive technique. But finding a genetic variant is not. That is where the whole problem is. So it's basically you have like millions of single nucleotide polymorphism in each of the individuals, and some of the effects that each of these variants shows are more cumulative in nature. It's not like, you know, it's a and the term that we actually like to use is called as penetrance. So we don't know what is the penetrance of a variant, and then if unless it is 100% penetrance, then and most of the variants they are not have that kind of a penetrance, and only variants which have those kind of penetrance are the one which are on the enzymes. So if you have a SNP which is within the enzyme and it's within the enzyme active pockets, like best example is sickle cell anemia, where you have that one SNP is actually valent to isoleucine SNP is in that hemoglobin binding pocket, and that means every single individual with that one SNP will actually give rise to sickle cell. So if you have a condition like that, which is mostly in case of biochemical enzymes that are active in the system, but apart from that, if any other transcription factor or any other kind of component of various component of cells, you will never get that kind of the penetrance in the population. And once you don't have that kind of a penetrance, then it's get harder and harder to classify their importance in the. Okay, and essentially you, Dr. Amit yesterday went through this, but then the idea here is that you can classify variants based on what kind of evidence is present in this, and then based on that you can use them for various purposes. Like you can use variants of strong clinical significance like tier 1 variants, and they can be, and for that those variants are used for therapeutic, prognostic as well as diagnostic purpose. And similarly, actually you can also use tier 2 variant, which is for these purposes. Again, they need also, you know, various level of evidences. But one of the most challenging one for this is basically for somatic variants, but then tier 3 variants, they are the most interesting ones, but that is when we don't have any handle to deal with, and they are variants of unknown clinical significance. And most of your patient samples, you will see a lot of them having a very strong association with the phenotype that you are seeing, but you have no idea why you are seeing them, or you need an extra layer of experimental evidence to prove that this. Like one of the example that I can actually think of, that we had a patient sample where we got a SNP in NeuroD, and this boy, it's a 14-year-old boy, and he has seizures. So, but then there is no way to prove that, or there was no literature even available that particular gene, NeuroD gene, any mutation in NeuroD gene has any seizure phenotype. So now the question is, but that is the only association that we can see. So now what we need to do. So what the group, our pediatrics group did, they deleted that they created the same deletion in zebrafish, and they found that zebrafish show the same seizure phenotype. So, but then think about creating such a line of zebrafish and then testing these phenotypes will take months. And when you are doing this for gene after gene after gene, or SNP after SNP after SNP, it's actually gets more challenging. So, though this is the most interesting group of variants that you will ever see, but they have there. And then your analysis will show you large number of variants which are likely benign in nature, and we don't know why some of those variants are, and then some of them shows very strong association also with them. Okay, so while we can use some of the SNPs for classification, but one of the strongest area where we can use some of this NGS based method is for pharmacogenomics. So old way of doing medicine in India, and especially in Arabic countries also, they have used and they have treated each human being as an individual human being, and they classify human, and in traditional Ayurvedic medicines they are classifying human being as Kapha, Pitta and Vayu in their nature. But actually I think that is, maybe a Kerala term for that. So what is really happening? You are not considering all human being as uniform in nature, and then treatment, you are treating not the, you are not treating disease, you are treating that person, and each person need a tailored approach to treat. But when modern medicine came, and especially evolution of modern medicine was mainly for pathogenic diseases, especially like antibiotic was the first major thing happened, and it was highly successful, because at that point you are not treating, and you don't have to treat a person, rather you have to treat a germ, like you have to kill some germ, virus or something like that. So it's highly effective in that case. But when it comes to lifestyle diseases like diabetes and hypertension, we have to go back to that old idea of treating a person. And why we need to do that? Because of this, like each human being has a different based on the polymorphism they have in various genes, they will show a different pharmacokinetics and pharmacodynamic properties against a drug. Okay, and so basically, what is pharmacogenomics is a study of inherited differences in inter-individual, like between individuals, about drug disposition and its effect. So same drug given to two different people will be reacting two different ways, and we are actually studying how this is happening. Right? So pharmacogenetics is basically looking at the genetic variation and how that is interacting with drug, and pharmacogenomics is basically looking at those in a population, and giving you a idea that how the population in general is behaving, and what are the multigenic determinants for the drug response. Okay. So one of the things that we know all the time, that one doctor prescribes a drug, and then it's actually effective for some people. Some people don't respond at all, and some people actually has serious side effect. The same medicine actually given to the 3 different people, and the question is like, why that is so? So this difference comes from 2 different aspects. One is called as drug uptake, and second is from the drug metabolism. So how the drug is taken by each individual and how that metabolized. So if your cell, the cell that actually taking the drug has many receptors, that means what happens? You have the drug, and all the drug will go and bind to that cell, and it will have a very strong reaction. Okay. If you have fewer receptor for the drug, you will have weaker reaction. If you have no receptor, the drug will not work. It will be just you are just swallowing the pill. And also, if you have a different receptor than the receptor that you need to have, in that case also your drug will not work. Okay. The same thing, the drug uptake also, if actually drug is normal uptake, then drug will work as it is actually intended to work. But if there is a decreased uptake of this for some reason, like you have some channel issue in your cell, then it may build up in your extracellular space, and that will create toxicity. The other aspect is like how drug is metabolized. If drug is actually metabolized at a normal rate, then the normal drug will be respond to the normal response. But if it actually it breaks very fast, then you need more drug. Like, so you need a higher dosage of that same drug to that person. And the third thing, if the slowdown, there's a slow breakdown, then you need to reduce the dose. So we need to know all these things. And I will show you some few examples where, and all of this aspect what I have just showed you, it's controlled through the genetics. And that means the SNPs in some of the genes that will define the response of those drugs, either at the metabolic level or at the level of intake. So this is about a medicine, abacavir, which is actually a reverse transcriptase inhibitor used for the AIDS patient. Okay. So the patient, whoever carries this allele, HLA-B5701 allele, they are at very high risk of hypersensitivity. So now what they are doing, they put this in the medicine label, that if you are prescribing this drug to a patient, first test for this allele and make sure like they don't have this, and then if the patient is positive for this allele, then go for an alternate medicine. So this is one such example where you are really looking at a SNP and making sure like if that SNP is not there, then only you prescribe that medicine. You cannot otherwise prescribe this. The other example is actually warfarin. Okay. So I can show you many such examples, but this is another example where in case of warfarin, if the variation is there in two different genes like CYP2 C9 genes or VKORC1 gene, either of these genes have a SNP, that means you should reduce the dosage level. So first example we have seen that you cannot use the drug for that patient at all with the presence of that SNP. But in this case, you are actually changing the. So both examples we have seen. In one example, we are completely excluding that patient from taking that medicine, while in this case, you are actually need to lower the dose. So based on your pharmacogenomics output, you will be able to tailor your prescription. You can tailor your dosage, and then you can come to. So any of these things now actually, I will go to CPEX a little bit later, a couple of slides from here. There are recommendations for various genes and what is that gene SNP relation with the medicine and what you need to do with that, and we'll look into that later. So what are actually the challenges for implementing pharmacogenomics? So one of the biggest challenges that translating this genetic information to clinical action. So we'll see like there are fewer genes only those who are reported with such thing. It's not that that interaction is only for those genes. It's just because it's not studied well. Then again you are adding an extra cost, because if you need to do this testing, that means somebody has to pay for that. And then we need to have some kind of a genotypic testing, high quality genotypic testing, and some of these things are actually more related with US, but again, the practical challenge is to translate this information, genotypic information into a pharmacogenomics information. Okay. There is a consortium called as Clinical Pharmacogenomics Implementation Consortium, which is called as CPIC. Okay. So the whole idea here is they want to actually standardize this format, report guidelines format, and then they want to actually provide a freely available peer-reviewed data for pharmacogenomics. And this consortium is an international effort. And what they do, they actually publishes the drug and which SNPs or the gene that you need to test. So up to 2021 data is available in their website. I haven't seen after that. So each of them, so you can see like each year they have new drugs, like in this case, CYP2 D6 for tamoxifen or like that. So they will actually tell you that even if any of this drug you are prescribing, you should actually test for that gene before you prescribing and look for the SNPs that may be determinant for or to decide the dosage. So based on that, and they have a detailed guidelines for each of this medicine and now it is actually for other. This is just a list, and you can go to that website, and then you can read more about this. Okay. So what is the benefit of one of the major benefit is like you can prescribe drugs more effectively and avoid prescribing over prescribing a drug. Okay, and then also you can also make patient adhere to the practices that you want to do and the right drug. It will improve the life quality, reduce the cost, because you gave a drug which is not working in that patient means like the patient will be having that drug for a couple of months, so you already wasted a lot of money on that and time, valuable time that is lost. And you can also revive some of the drugs that failed the drug trial, because it might happen that they need to test that population for those SNP and the drug failed just because those things were not considered. So you can actually revive some of them, and of course you can control the cost. Yeah. So what are the potential legal and ethical issues? One of the major ethical issue is that who should have access to the person's genetic profile, because you are doing a genetic testing, so who will control that data? How will they protect your genetic privacy and prevent genetic discrimination in the workspace and in our healthcare system? So I usually try to joke when I teach in US that in India, we usually have arranged marriages, which is not much known to. And if somebody has access to my genetic data and say like I will have a heart attack at the age of 40, most of the people will not give me a girl for me. So, so that is so the whole idea is that how you can maintain the privacy of the data and then who will have access to it. Okay. And other thing is like your employer. Again, the private companies and all, when you're going and they know that, okay, you may have a tendency for depression or something like that, will they take into account when they are giving you a job? So all these kind of issues like privacy related issues and who will have access to that data, and even insurance. Now insurance is not that popular yet in India, but then it's getting popular. So insurance will classify this as a pre-existing condition. So will they pay you for some of the disease? So all these things actually is important that we have to think about when we are, again, this is more to do with US, but then one of the areas that which we have real problem is this integration of pharmacogenomics with the personalized medicine, and marry both of these things and make clinician aware of use of it. Okay. So now we are actually coming to the last part of my talk, which is we are moving to the 4th generation sequencing, and Dr. Amit actually already gave you a primer for this, that single cell analysis. The 4th generation of sequencing it's not a new sequencer, rather than it's more about a new way of doing things. Okay. So what are you doing here? You have a tissue. In bulk analysis, you just make a smoothie out of it, and then you analyze it, and then single cell analysis, you will analyze each cell separately, and you get the data out of it. So why it is important? Because if in India you go to a smoothie shop and you have a smoothie or mixed fruit juice, a lot of places have very popular. So the taste of the all mixed fruit juice is like, I'm from Hyderabad. Those people who are from Hyderabad, they will know. This is a very big delicacy in Hyderabad, all mixed fruit juice. So the taste of all mixed fruit juice will be according to what is the cheapest fruit in that part of the time, because and most of the time it will be banana. So you won't get even strawberry taste or anything. So essentially what I'm trying to say here is that in bulk RNA-seq, the flavor that you get out of your data more or less coming from the dominant cell type or dominant the phenotype that you are. But the whole idea is that if that is what we are looking for, it's fine. At the end of the day, we just want everything else a little bit, but then something dominant, we are fine with that, then it's okay. So that one limitation is something that we can actually overcome using single cell RNA. But why we actually need this? So one of the best example is cancer. So we always think when we think about cancer, every type of cancer we think as a homogeneous mass, like something proliferating, and then everything is fine. But that is not the case. And the main thing what we learn over a period of time is that it's more heterogeneous. The cells, they are in the core, the center of the mass cancer versus something that is actually exposed to the periphery. And those who are nearby the vascular system, like where the angiogenesis is happening, they all are different. So which means that there is an inherent heterogeneity, and that is why a single drug cannot target all the cell in even the chemotherapy and all, and you still left with some cells in the system. Okay. So if you want to design a drug or a treatment strategy, you need to understand the heterogeneity of your tumor, and you need to actually address the heterogeneity part of that, so that all the cells will be equally targeted, or at least you have a combination of approach that can actually target everything. Okay. So knowing heterogeneity is important, and same thing actually goes to various other areas where when you are looking at tissue homeostasis, or you are looking at what are the different cell types in the tissue and how they are actually interacting at any given point of time. So heterogeneity is the rule in biology, and without taking that into account, we cannot actually make sense out of the system. So to understand heterogeneity, we need to do single cell rather than going for the bulk RNA. So this is how actually it is. You take something like a PMBSE or anything you are taking, you can basically generate the catalog of all different cell types, and also you can actually get some sense of the population dynamics of those cells, how they are changing their nature. So, single cell RNA-seq can be done with 3 different strategies. Okay. So, one is called as single barcoding, second is called as split pool barcoding, and third is through microfluidic space system. Okay. And I know that you all are familiar with all 3 when I actually show you what it is. In single barcoding, what you do, you sort your cell into one cell into each well and add one barcode, do an lyse the cell there, do the RT-PCR. In that RT-PCR step, you are adding one index into that. So, that will give you unique identity to each cell. And then you just after that doing that RT, you combine everything and then do a library prep and sequencing. So, then what is happening, each cell has a barcode. So, the RNA that is coming from each of those cells will have that barcode. So, you use that barcode and then just deconvolute later on. But the problem with this approach is this is very low throughput, because you cannot analyze more than 90 or 100 cells or 200 cells through this method, and it is expensive also, because you are doing one well at a time, which makes it very expensive. Okay. But there are systems like iCell8 and those kind of systems are there, which are essentially doing symptoms similar to this. Okay. But this is again, this is not the way single cell technology is going. We are not actually not much into this kind of approach, because the cost become cost prohibitive. Yeah, it started this way, but now it's not that popular. For the historical context, I thought it will be it's a good idea to start this way. So, this other thing what we do is called as split pool barcoding strategy. Okay. So, split pool barcoding, what we are doing, if suppose you have you want to analyze 10,000 cells. Okay. Now, you pool 100 cells and split them into 100 wells. So, you have 100 cells, you split them into 100 wells. Do first RT. So, each well has a barcode. So, that means all the 100 cells will get one same barcode. Now, once you have barcoded, you have did your first RT, you pull every cell back, all the 10,000. So, now 100 cells in 100 wells, you pull it back, you have 10,000 cells back into one pool. You split it again into 100 cells. So, now and then put it into 100 wells. So, now each well will have a different combination of cells. You add your second barcode. So, that means each of those 100 cells you got a different combination of barcode now. Then you pull again and then you split again. And if you do this split pool barcoding 3 times with all the available barcoding, you will generate approximately 100,000 combinations. And that 100,000 combination you will actually and then pull everything and then make a sequence. So, basically what you are doing, you will each of this cell will have this, like you have this is of like 4 set of barcode, and this also have 4, but you see the order of each of these barcodes are different. So, it is the combination of barcodes, also the order of those barcodes are also different. Yeah. So, this way you can create any number of combination through just by splitting and pulling. So, what is the advantage of this system is you do not need any specialized instrument, because it is just pipetting based. You have, say you have 100,000 10,000 cells in 1 ml of media, 1 ml of media, you actually split them into 100 microliters into each, that 10 microliters into each, that means each of them will have 100 cells.
How do we extrapolate which cell is this? Suppose this is monocyte or this is...
That is based on your transcriptome. So, you will be, so it is only the barcoding part I am talking about.
So what is this barcoding is?
Coding
Actually.
Barcoding is actually, so you have a RNA, okay, you are putting a tag in front of that RNA to identify that. It's a string of nucleotide only, but it is actually, so you are putting that in front of an RNA, and if you have say 10 different genes coming from the same cell, all of them will have same string of DNA. So, then you will, so the first thing is like based on that you will combine all your RNA data into and say like this is coming from cell number 1, this is coming from cell number 2, this is coming from. Yeah, they already have that. So it is called as.
How does it compare with?
It is good. We actually started offering that from our service. Called as. So we. Okay, I will come to that. Just let me finish this and then come. So this is actually called as. So this is called a split pool barcoding strategy. The whole single cell analysis is about barcoding. How you barcode a cell transcript. Okay. So first way of doing is like you pipette cells. Second way is like this split pool strategy, and you create a combination of barcodes inside each cell. The third one is through microfluidics, which I will be saying. Now coming back to the PIP-seq. So we were the lab which was providing this for them. So they were providing initially services. A company called as Fluent who generated this system called as PIP-seq, and it's again based on split pool barcoding. But now Illumina purchased that platform. So we were the one who was sequencing from last 2-3 years, and we are offering that as a service from YCGA now. So it's simple. You actually take cells, put into a mixture of things, vortex it. They have a vortexer, and then you spin it down, and then they have some pipetting things. That's it. So it's very straightforward and it works well. I will actually send you a paper. There is a benchmarking paper is there. Our own benchmarking also shows that it's comparable with the 10x. So it's comparable with 10x data. Very cheap actually. It's almost 4 times cheaper than. What specificity?
She's asking if you are putting 3 or 4 tags in front of one piece of RNA.
No, so you are putting them one at a time. So that means that combinatorial. And each of them, yeah, will be having unique combination. So that is how you get that specificity.
Like my cell population is a TG entry, then I will be putting barcodes for monocytes or.
No, it has nothing to do. It's way before monocyte or anything. Forget about that. It's just cells are cells. Okay. What we are, whether they are monocytes or not, that information will come from the gene expression data, not from the barcode data. Okay. We are just saying cell number 1, cell number 2. So you need to identify that if you are sequencing a piece of RNA, whether it is coming from cell number 1 or cell number 2 or cell number 3. So to classify that, you are doing this barcoding. Okay. And once you get that, then you have the RNA. So you will get all the RNA coming from cell number 1. Now you go and classify that cell based on the RNA content, that whether this is a monocyte, this is a T cell, native T cell, CD44, all these things.
But within a seemingly homogeneous population, even like a monocyte, there will be different types.
Yeah, there will be different types are there. All those, so you can do all those things. So essentially the barcoding step is to barcode a cell. And
Barcode is cell. How can this specific barcode be added inside a cell?
when you do RT-PCR, that is what you're doing.
So the cell should be live.
So in the case of
Yeah. So at some stage they are actually doing the lysis. Okay. So in split pool strategy, they usually what they are doing, they are actually lyse at the last, but then the reagents can actually go inside the cell. They are made permeabilized and then they go inside the cell, and you can do in situ RT-PCRs. That is what actually essentially they are doing. Then you don't have to lyse. Lysis happens after all the barcoding steps are done.
So cells are initially permeabilized and then they just go inside and
Yeah. Yeah, because you need to maintain the integrity of the cells at that point.
So we should know the cell number. Is it specific to cell number, the barcode?
No, so parse, what Mr. was talking about. So parse is actually, you can do, the current kit you can do up to 100,000 cell barcoding. They have a kit for that. So yes, all these barcoding kits actually they comes with for the specific number, like they have 10,000, 20,000, 100,000. So you have to, based on what number you are targeting, you need to actually start with that kit, and there is always a recommendation for how many cells. So if you want to capture 10,000 cells, you cannot start with 10,000 cells. You need more than that. So like PIP-seq, you need at least 2 times more than that. If you want to capture 10,000 cells, your input cell number will be 20,000 cells, and then only you will. So capture efficiency, what the term that used here is called as capture efficiency, and for 10x it is around 65%. But for other like parse and Fluent, it is actually around 50%. So based on capture efficiency, you need to extrapolate. So if it is something having a 65% capture efficiency, that means if you want to capture 10,000 cells, then your input will be 17,000 cells. If something has a 50,000 capture efficiency, then you will have to have a 20,000 cells. So like that. So and then again there is another thing actually. I just okay. Let me finish it, and then I will come back to another round of discussion on something else. So in case of microfluidics, again the idea here we are still barcoding cells, okay, and then we are doing rest of it. So in this case, what we are doing, there is a bead, okay. This bead consists of a adapter, and each bead will have a unique barcode. Each bead will have a unique barcode, and then some other portions will be there. Then what you do, you send it through a microfluidic chamber, and it has 3 inlets. From one side your bead will be coming, from other sides the reagents and beads will come, other side cell will come, and from the third side the pressure inlet and oil is actually going, and a pressure is exerted. In this process what happens, in this junction where these inlets are coming, they make a oil droplet, and this pressure is controlled in such a way that each oil droplet will have one bead, one cell, and all the reagents required for RT-PCR. And then this, what they do inside that oil droplet, it's like an
Might not be the case actually. I mean, like a lot of wells, a lot of droplets will be there where there might not be cells, where there might be two cells.
yeah.
Everything will not be
No. That's where I will come to that, like how that is done. So basically, as a workflow, what happens that you have. So idea is that you get a oil droplet, and each oil droplet will have one cell, and each droplet should have some reagent and one bead. Okay, and now the reagents are there in this. They will lyse cell inside that oil droplet and do an RT-PCR, which will be so in that RT-PCR step, the primers are provided by that bead. So that means it will insert a barcode into your cDNA. Now after that, what you do, you break open the oil droplet. Okay, you add a detergent that will break open and then pull your whole RNA now together and just sequence it. Now what Amit sir was talking about, the innovation of a technology is there are 2 issues in this case. One is called as doublet rate, where you are actually having 2 or 3 cells going into single oil droplet. It never happens. Actually, mostly it's the 2, 3 rarely happens. It's actually, if you are that bad in your experimenting, then problem. So, and dropout rates. So there will be some oil droplet where there is no cell. Okay. So either of these conditions. So one of the way it is done is through controlling the cell number that you are, and if you have a good suspension, you have cells are single cell suspension, and they are not sticky or anything like that. That is how it is done. The second thing, the whole innovation is in the pressure, the control of the pressure by the instrument, the chromium system or anything. That is one of the other factors here. But the third thing, which is basically relevant for doublet rate, is that more cells you analyze. Like say if you are analyzing 5,000 cells, you will get fewer doublets. But doublet rate increases exponentially as you analyze more number of cells per channel. So suppose so the doublet rate increases to almost 7% when you are going from 5,000 to 10,000 cells per channel. So, but then there are bioinformatics way. Hopefully, Francis will be showing you that how to remove the doublets based on. No, you are not showing that filtering. Okay, so you can actually bioinformatically you can filter the doublets from your data, and then you can clean up it for that. So that is how the doublets are taken care of. Okay. Yeah. We are just introducing them to this. So again, why single cell? Basically, we are cataloging different cell types. We want to identify new cell types. We want to identify differentiation trajectory or dynamics of any developmental process. Yes. Okay. But what are the biggest challenge for single cell analysis? One of the biggest challenges, it's big data set. Okay, it's so big. The other challenge is shallow sequencing. So most of the samples are sequenced at, say, every cell is sequenced for 20,000 reads. So that is the minimum sequencing. Some people go all the way up to 50,000. But think about this, on an average, 1,000 genes are expressed per cell. That means you only have 2 or 3 reads per cell, per gene in your data, and how reliable that shallow sequencing is. So this is something that you always think about when you make quantitative inferences from single cell data. I always try to argue with people that single cell data is non-quantitative. It is more like a qualitative data. So please treat that as a qualitative data. Don't use that for gene expression. And then because of that, like you are shallow sequencing, and each cell has its own variability, the technical variability as well as biological variability of data is very high. Okay. So one of the best practices, because of these issues, because of high technical variability as well as the biological variability, do not confound your experiment by batches. If you have one set of experiment you are doing, don't do them in batches rather than do everything together, because that will make it easier for you to. And if you are actually doing in batches, then split your replicates in batches. So don't do like for one set of sample you do all replicates today, and then other set of batch you do other replicates. Rather than that, split across the sample replicates in different batches. That will also help you to, and add as many replicates as possible. And this is easier than said. The biggest problem is the cost. So even like RNA-seq, people are reluctant to add more replicates. Here actually you are talking about 10 times more cost than the bulk RNA-seq, and so it's a harder thing to do. And also whatever actually Francis told you about the RNA-seq, it's also relevant that always add metadata or collect all the metadata about your experiment, and that will help us to understand the batch effects. So technical variability of most of the RNA-seq experiments comes from cell-specific capture efficiency, library quality, amplification bias, and the batch effect. So one point I want to actually emphasize here about amplification bias. Why amplification bias happens? Specifically, it was mentioned in not the bulk RNA-seq, but it is being mentioned in case of single cell RNA-seq. The reason why this happens, because you are working with a small input material. Any small input library prep, DNA, RNA, all of them will show some level of amplification bias. Okay, and we see a dominant effect of amplification bias in single cell libraries because of that. And the source of biological variability comes from transcriptional bursting. Anybody know what is this transcriptional bursting means? Okay, none of you are molecular biologists, okay. So I'm happy that I can even say something wrong. Okay, so what do you mean by transcriptional bursting? So tell me, when you think about transcription, what do you think, how it happens?
As long as the replication is going, the transcription is getting slow.
So that is what I also thought, okay. That your RNA polymerase comes and transcribes, and then another polymerase will come and transcribe, and another polymerase will come and transcribe. But in reality, that is not how it happens. It happens in bursts. So polymerase will come and engage for 30 seconds or 1 minute, and it will transcribe everything, and then everything is silent. Okay, then like after 5 minutes, again it will happen, and it will happen again after 5 minutes or 3 minutes or 2 minutes. So it happens in burst, and this is called as transcriptional burst. If you actually ever quantify a nascent RNA on a microscope based method or imaging based method, you will be able to see that transcriptional burst happening. So the idea that transcription happens as a continuous process is not true. Transcription happens in burst, and then burst that is precisely controlled process. So
Single cell will not be able to see that burst.
no, that is the problem. The single cell, it actually taking that snapshot more effectively. In the bulk RNA-seq, you are actually normalizing that because you have millions of cells, and now say if you have a million cell in your soup, 10,000 cells will be bursting, 20,000 cells are not bursting. So you will not be able to see the effect of burst, but in single cell, you are picking that one cell, and then you are actually putting it through your active. So that is where the cell is same. These two cells are same, but one is not bursting, one is bursting. Then it will be seen as two. So that is the source of biological and you are. So every cell, every gene have do that. That is how the transcription happens for every single gene. That is a mechanism. So single cell is actually able to capture that. That's why you see a lot of biological variability in single cell data compared to bulk. Then again, rate of RNA processing. So each gene actually processed at a different rate. So the one which is processed slightly earlier versus late. All this dynamics of transcription and processing is captured through the single cell.
Even for genes where there is only slight difference where they can pick on bulk, the same difference would it be exaggerated on a single cell?
Yeah, and it has to be controlled. So the whole this biological variability thing coming from singles. Sometimes you see clusters, cells getting clustered into two different clusters, and you see more or less their general expression profiles are same. That is coming from this kind of changes. It's not a real difference. Rather than it's more like coming from all this kind of thing, which is actually giving rise to the biological variability. Okay. So then, the single cell is good, but the biggest challenge, like we started with heterogeneity, okay, the whole idea of heterogeneity, just knowing that you have a cell doesn't matter. You need to know where is that cell, what is the position of that cell, okay. And think about a neighbor, you have a neighbor who are like very rowdy kind of a person, like, or the morning, everyday morning, or you have a house next to your house and husband and wife is fighting every day morning, and that is what you are hearing every day when you wake up. So your days are spoiled because of your neighbor. It's not because of you, but because of your neighbor.
I actually don't live close to
and same thing, but think about a very nice person, morning you actually come out to pick your newspaper and this guy say, hey, hi, how are you doing? Well, that makes your day. Huh? So neighborhood is actually one of the key determinant for disease development, everything. Cells, those are close to vasculature, they behave differently than those which are far from the like blood vessels. Or cells, those are in anaerobic environment, not anaerobic actually, but there's oxygen related term, I forgot that. What is that? Yeah, whatever it is. So basically the area where you have less oxygen tension, it's not hypoxic, actually they have anoxic or something, it's anaeroxic or something, that is the word. Basic idea is that wherever you have less oxygen tension, cells behave differently. And you are further from the vasculature or the vein or the artery, the tissue will also behave slightly differently. So you need to know your neighbor, and knowing the neighbor is important. And that is when you actually do spatially resolved single cell multiomics. So this is a word that it's a right word. When we say spatial technology, rather we like to say spatially resolved single cell sequence. So you want to see single cell data, but you also need to have a spatial information along with the data. Okay, and you can do that two different ways. Something like called as sequencing based, and other is actually imaging based technology. Oh, I know why my slides are behaving this way. I have to change my resolution of my laptop. I don't know. That's okay. Anyway, so sequencing based method, what you are doing, you have somehow you are barcoding so you are here. You are including two barcodes. One barcode for the cell, because you need to identify each of the cells separately, and then you also need another barcode to identify the relative location of that cell to compare to each other. So you need to introduce those two barcodes, and then you do a sequencing. But then also you can do that based on imaging based approaches. So example of imaging based technologies like Visium, Cosmex, Xenium, like that. And if you are thinking about sequencing based, you have again Visium or Geomax or Curio. So some of these platforms give you, and we'll see some of this quickly, what it does. So the biggest challenge in the spatial technology field is the trade-off between resolution and number of targets, because these are expensive, and if you add more number of targets in your analysis, it will cost you more. Okay. The workflows for most of the spatial technology is hard, it's complex. And you need to optimize each tissue, especially spatial imaging based spatial platform. You need to do clearing, because there will be a lot of autofluorescence coming from the tissue, and you need to have a means to clear them. or the permeabilization protocol. So, each of this tissue for barcoding, effective barcoding, you need to permeabilize tissue properly, and each tissues with based on their fiber content as well as like how much cartilage or bony contents are there, based on that you need to actually change your clearing as well as permeabilization protocol. Then high assay to assay variability. So, this is the field newer field, and we do not know what is the real source of this. It may be biological in nature because you have no means to try same tissue section 2 times. So, we do not know that is a technical variation or is it coming from because if you are taking 2 sections, they are not same, they are actually different because they are 2 different layers. So, that means we do not know where this is coming off. Again, the assay cost is a big thing. The other issue is, are you actually interested to do discovery? Discovery means like you need to test as many as genes, or you want to have a focus question like neuro-oncology panel for cancer or something like. So, the whole idea is a discovery versus focus question. Then it actually is expensive. These instruments are expensive, so you need to spend additional money for capital equipment, and definitely there is a big bottleneck in terms of visualization, data analysis and those kind of things, because there are not many people who are expert of doing. Okay. So, the first very popular what is happening? It copied multiple times. Francis, what is happening? This is your laptop, it's not behaving, it's like you, not behaving properly.
No, it's the other people scrolls in a different way. That
He doesn't like you. His laptop doesn't.
everybody's wrong except me. That's what I'm thinking.
My boss actually used to say that.
Bonnie, you don't know how to scroll in the other way.
I'm not. Oh, no, it's not going anyway. See, it's coming back. What to do? Something is.
Something going on. What did you do to my computer? Are you pressing it?
They could finish it. Done.
Some people it worked. For some people it is working.
It still is not. It's running.
For some people it has, but not for everyone.
Not for everyone. I sent an email to AWS. Yeah, one we could check.
So, okay. So, the first platform, which is it's called Curio, and I want to. This is a sequencing based platform. So, what are you doing here? This is a small slide, okay, and on the small slide there is a lawn of oligos, which are barcodes, so spatial barcodes, okay, and then what you are doing, you lay your section. It's actually 5 to 10 micron sections. You lay your sections, and mostly we just got the FFP protocol, but it was earlier just the fresh frozen sections, OCT embedded sections. You put the sections over that area here. So this is a lawn of oligos, and this consists of this set of barcodes there and a bead. Okay. Now you put your sections over that and put your finger at the bottom of the slide. The idea is that we have a body temperature of about 37 degrees centigrade. So we just allow that section to melt over that region. So just do that for like a couple of seconds and then melt it, and then you do a RT-PCR on, and there's a permeabilization step, and do RT-PCR, collect your RNA and just sequence it. It's a very simple, straightforward technique, but so this is actually called as curio. Okay.
But how many genes can it do?
It's a whole transcriptome. So it comes in two different variation. You have either you can analyze 3 by 3 sections, 3 millimeter by 3 millimeter section, or you can analyze 10 millimeter by 10 millimeter section. And the advantage, because this is sequencing based, so you are sequencing whole transcriptome, you are analyzing all the and the beauty of this method, which you will understand why I am saying this, because you can, it's a species agnostic. Means you can analyze any species using this, because it's not looking at it's like a sequencing method where after sequencing you align it to the reference genome and then you figure out that what it is. Okay, so this is curio, and here is an example of data. This is a retinal sample from mouse retina, and you can see expression of two genes. One is this GFAP and Rho. So Rho is basically present in photoreceptor 1. Yeah. So that is actually present in photoreceptor 1 layer, photoreceptor layer, and you can see the expression of that in your retina, and also you can see GFAP expression in optic nerve head. So you can really see a granular data, but the resolution for this method is 10 micron. So that means if a cell is less than 10 micron, and then it will not be actually able to resolve those cells. But then this kind of analysis is very useful for neighborhood characterization and general understanding of tissue. They came with a new technique called as tracker, the curio tracker, and which gives you a data at single cell resolution. And at this point, in all sequencing method, curio tracker is the only platform which can give you single cell resolution spatial data. Okay. I don't know what happened to my slides. Actually, everything got messed up. Okay. So next one is an imaging based technology called as Cosmex. So this is developed by a company called as NanoString, and the way this assay works, you create a custom set of probes for your whatever number of genes you want to analyze. Say you want to analyze 50 genes. So you decide which 50 genes you want, and you generate the probes against those 50 genes. Okay. And the probes are usually 20 base pair probes, and this probe will go and bind again, hybridize against. So you have a slide, and then you go and hybridize that probe, and then what happens that you do something called as amplify the signature using branched PCR reaction. So it's actually every PCR cycle it will add one extra floor. That is what actually happening, and so that is how you increase the concentration of floor, and you do this in a combinatorial manner. So you have multiple probes for each gene. So there are 5 probes for each gene, but all 5 probes you will not add in each cycle. So each cycle you will have a set of probes for set of genes, and then you are actually hybridize it, do the amplification, take a picture, wash it off or quench it, and then you actually come back and you add next round of it. So that way what happen, you will, so why you are doing this? Any idea why this way? You can actually just add all the probes together. Why you are doing that? Any idea? Because these are 20 nucleotide probe. The chance is that they may be binding to a non-specific target. So how you will get those specificity? If you have 5 oligos and each of them added a different cycle, and at the same spot they illuminate, that means you know that is a specific one. But in random one, sometime you get a signal, you omit them from your analysis. So that is how you generate the specificity here. So this is called as Cosmex. Cosmex is the only imaging platform at this point which can give you whole transcriptome data. So, this is how this works. Now, again they have a slide, and this area you can put as many samples as you want, and then you can see localization of genes at subcellular levels also. You can actually see that because they are using DAPI, and there is another idea in spatial technology called as segmentation. So, cell segmentation is a very challenging problem in spatial technologies. Cosmex, they uses a mixture of 5 cell surface markers, and based on their presence they actually mark the boundary of cells, and then they use but they also use a computational approach to mark those boundaries, make sure that all the cells. So, some cells and then they use DAPI. So, one of the biggest challenge, like think about cell like muscle have multiple nucleus. So, that means if your DAPI stain is coming, that will show you a string of, but none of those DAPI stains are actually able to show the boundary of cell, because the cell boundary may be far off. Same way like neurons have a soma, and it has like all the body going all the way to like kilometers. So those kind of cells, it's sometimes hard when you act, some of the computational programs are hard to work on, but the things are improving, and so segmentation is one of the major challenges for many of these technologies. Okay. Other technology which is from 10x called as Xenium. Here they use padlock probes. What is a padlock probes? So, padlock probes, it's same thing what we are talking about in case of Olink we said. So, you have instead of antibody, you think about two oligos, 2 primers. They are at proximity to each other. So, they will go and hybridize, and they will bind next to each other, and there is a DNA molecule having complementarity to each other. So, that means you will actually amplify that, and each amplification step you add a fluorophore. So, when that amplification happens, that signal will be coming out, and that signal you will capture through imaging platform. So, the specificity comes from 2 probes each gene they are using, because then what is happening, binding 2 non-specific fragment with the same complementary DNA is actually not possible. So, that way you will get a very high and here actually before doing the amplification, they padlock probes, they ligate to each other and make that circle structure, and they do a rolling circle amplification. That is how they achieve this. And here again, this is an example how mouse brain, this is our own data, where you can see the position of various cell type in a tissue, as well as you can see their single cell distribution. This is a UMAP, a TSNE plot, and through this you can actually see various cells happening. And this is one example of how you do a computation based segmentation. So, here they are not using cell surface marker, rather than what they are doing, they are looking at the DAPI signal, and then they are expanding the DAPI signal or to all the sides for up to 15 micron, or before all the cells are expanding, if some other cells actually come before 15 micron, they will stop there and draw a boundary, and that is how they make a cell to cell boundary prediction using computational based segmentation. This is one example, and here cells are colored, pseudo colored based on the cluster they are classified, and the gene expression is also shown in this case. Okay, so this is some of the way you can do this. You can also do spatial epigenetics. So, it is not just like you can study expression, but you can also look at chromatin accessibility or protein DNA interactions, like cut and tag you can do on this, or like chip experiments you can do. And this platform is called as spatial epigenetics platform, called as Atlaxomics. So, what they are doing here, you put your tissue and then flow barcode. First you flow one set of barcode in this direction, and then you turn your slide and flow the second direction 90 degree perpendicular to it. And doing that flowing of barcode in 2 different directions, what it gives you is a combination of barcodes over the tissue, and then you go and do your experiment on that. So, essentially you can do spatial ATAC-seq and cut and tag. How many people know what is cut and tag? Anybody know? Okay. How many of you know what is chip-seq? So, chip-seq is to study histone modifications, DNA protein interactions and all. Cut and tag is actually chip-seq, but it actually uses a tagmentation based instead of sonication, or sharing is done through tagmentation. So, that makes it easier, and you can do with a smaller number of cells. Okay. So, So you can do spatial ataxic and spatial cut and tag on this, and this where you are essentially, I just mentioned you, like you will flow barcode for one end to other end, and then you flow for the other direction, and this will generate a combinatorial barcoding, and then you use that for you make library out of it and just sequence it. So this is how it looks like. So I think I will just stop at this this point. So I just want to add one more quick thing, and then this I will stop. So one of the main, how many cells you need to analyze for a single cell, and how you actually decide that. Because you will ask people like, this is a question that I always get. Like people when they are asking for some kind of a consultation, they will say, hey, I want how many cells I should be assaying. So the number of cells that you need to assay depends upon the frequency of your cell in your population. Suppose you want to analyze a cell, or you want to see any cell type which is represented as low as 1%. So if this is a number that you are thinking about, that okay, I want anything that is around 1% of the population, I want to see that number of cells. You need at least 50 cells for a statistically relevant cluster. Okay. So most of the programs that make cluster, they can actually make cluster out of 20 cells, but it's always good to have at least 50 cells in a cluster. Now, so if you need a 50 cells in a cluster, and your cell is represented only 1% of time, that means you need at least 5,000 cells for your analysis. Okay. But suppose you only interested in those cells which are more than 10% representation, that means even if you analyze 500 cells, you will get that number. But if you are looking for 0.01%, like very rare cell type, that means now you have to sequence almost 50,000 or 500,000 cells to get that rare population. So it's not like everybody you will see like 10,000 cells. 10,000 cells has no meaning. The number of cells, how many you need to analyze, depends upon how rare the cell population that you want to analyze, and this is how you calculate that number. Okay. So I think we have covered extensively, we have looked at almost everything, and just one other aspect. Nowadays, sequencing technologies and sequencers are moving away from, you can close it, we actually moving away from just from the sequencing application. So if you think about sequencer is nothing but a high speed camera. Okay, it's a microscope with high speed camera. So now what they are doing, they are using flow cell to image cell in the sequencer, and the AVT sequencer I have showed you from the element, it can actually do cell morphology analysis, in situ RNA sequencing, then protein analysis on the flow cell. So you put your cells on the flow cell itself, and it will image from there, because these are camera, high speed camera with a microscope. So the flow cells and the sequencers are actually opening up new applications, such as multiomics analysis on the flow cells without sequencing, imaging based applications. So the things are changing, new applications are being developed, new capabilities are unlocked on these flow cells, and sequencers are actually moving from just a element of sequencing, but rather than they are actually becoming more and more instrument to study multiomics, like cell morphology, protein abundance, in situ sequencing, perturbation analysis on scale. So things are actually changing, and this is the most exciting time for genomics. Okay. So with that, I think I'm done with my next-gen sequencing classes, and I'm here for next 2 days. So any questions, feel free to ask. I'm happy to.
So we are saying single cell sequencing with PARSI and 10x as well. So the major problem we are facing that the fixation of the cells. So in PARSI kit, they told us we are usually trying to fix the cells, and after fixation the viability is very less. In the 10x kit, they told us the viability should be more than 80%. So
how fixed cells will not be viable? How fixed cells will be viable?
So in 10x case, the cells is viable.
No, the moment you fix a cell should die. So usually the fixation protocol, they have like something called as methanol fixation. That is what they use. The moment you have fixed cells, you don't need that viability numbers need not be there. Before fixing, the cell viability needs to be 80% or 90%. Okay, because after fixing, cells are dead. You cannot actually Huh?
So that means integrity they maintain
Cell integrity is maintained. The cell membranes are that is what they are looking for. But then, if the moment you fix anything, that means they are dead. They cannot be.
But sir, 10x it the fixation the biology is maintained. So we have already.
It cannot. How? The whole idea of fixation itself is to kill the cell.
Even those cases.
I had a question about like heterogeneity. So like you mentioned that